Next up, we have Biljana from the Fraunhofer Institute for Cell Therapy and Immunology. She's studying for a master's in biomedical sciences, and her focus is on cell biology, microbiology, genetics, protein analysis, and pharmacy. She's currently also working at the Department of Microdiagnostics at the Fraunhofer, where she's completed her master's and was introduced to the breast research field. Over to you. Thank you.
On behalf of our team of the Fraunhofer Institute for Cell Therapy and Immunology in Leipzig, Germany, I would like to welcome you at the Breath Biopsy Conference 2022. This presentation shows our work in the field of breath gas analysis by the topic of the identification of COVID-19 marker in exhaled breath by the use of the ion mobility spectrometry. The human expirate, which is a composition of individual components consisting among others oxygen, carbon dioxide and other metabolites. Due to the constant exchange of endo and exogenous processes, pathogens can invade human organism. Via the bloodstream, they enter the cells and metabolize in each case and results volatile organic compounds. In terms of diagnostics, VOC compositions and quantity can vary and eventually accumulate in the expirate. These variations can be applied in diagnostics for data analysis to differentiate between physiological from pathophysiological conditions. The following work serves as a model for the analysis of human breath by the detection of specific VOCs as potential markers. The current epidemic of SARS-CoV-2 has demonstrated the importance of rapid diagnostic procedures in infectious diseases to detect infected individuals and subsequently isolate them to break chains of infections. From the beginning, tests based on PCR assays have been the gold standard. However, due to their higher cost and laboratory complexity, they are not suitable for mass screenings by the populations. Lateral flow tests have established themselves as screening methods for rapid detection of SARS-CoV-2 antigens, which are easy to perform, inexpensive to produce, and provide a result within 10 to 30 minutes. However, the sensitivity and specificity of antigen tests are lower than that of PCR assays. Studies describing the use of successful olfactory identification of SARS-CoV-2 infected individuals, which have been conducted since the beginning of the epidemic. They identified by the use of exhaled VOCs out of the odor with a relatively high sensitivity and specificity. From our point of view, GC-IMS technology could be an additional method next to the incumbent antigen tests in the future. Especially the support of screening specific population groups such as nursing homes, airline passengers, or company employees. For this purpose, the method should be as fast and uncomplicated as possible and show very few false negative and false positive results. The present feasibility study aimed to investigate whether a simplified sampling of exhaled air can distinguish SARS-CoV-2 infected subjects from non-infected ones at an early stage after the infection. The ion mobility spectrometry is a highly sensitive physicochemical analysis method for detecting substances in a gas phase. It is used also in the fields of forensics as well as in the diagnostic research. The principle of IMS is the separation of ionized molecules, which are separated in an electric field according to their mass-to-charge ion mobility. By coupling a GC column with an IMS, the separation capabilities of both devices are combined, so that complex sample mixtures can be more specific identified. Separation takes place in two separate compartments. The breath gets pumped into the sampling tube and thereby transported into the GC IMS. In the GC, the sample mixture is pre-separated, whereby the analytes are separated based on their specific retention time in seconds at 60 degrees Celsius. Afterwards, the separated molecules are transported into the IMS through the carrier gas of the ambient air into the ionization zone. There the molecules interact with the ionization source of tritium and get ionized. These collide here in a specific drift velocity along the drift zone depending on their mass, charge, charge distribution, and structure. The connections also occur here by collision, which ionizes the product ion and is detected as an analyte peak at the collector. The analyte peaks detected at different times provide informations regarding the specific drift times and consequently the assignment of the analyte in a sample mixture. The data analysis is shown here schematically, which included the creation of heat maps for the visual representation of the detected analyte peaks of the GC IMS measurements out of its automatically visualized chromatograms for each recorded second. Afterwards, followed by the cluster analysis, which first checks the matching and non-matching features out of the datas, and then forming matching features as clusters. In conclusion, the statistical verification of the determined clusters was performed by the use of the linear discriminant analysis for the classification. The recruiting and sampling for this work took place by the medical practice of Lip and and the patients who were PCR tested for suspected COVID-19 disease. Those who agreed to the study were given a FFP2 mask and freshly unwrapped sampling tube to place under the mask in front of the nose-mouth area as shown in Figure 1. The GC-IMS measurements were performed by the practice staff using the step sensor technique directly in the patient's room. The sample took place by pumping the breath into the GC-IMS by the internal pump. The analysis took place at ambient pressure. The GC-IMS visualized the signal intensity at the detector in volt as a function of IMS drift time in microseconds and was recorded over a total duration of 209 seconds. Nasopharyngeal swab samples were taken for PCR tests and were assigned to the subjects. 79 PCR-positive patients served as SARS-CoV-2 positive subjects and 44 PCR negative patients as SARS-CoV negative subjects. The PCR results obtained from the nasopharyngeal swabs according to the duration since symptoms onset with a wide spread of CT values. However, a total of samples from 123 subjects with COVID-19-like symptoms were analyzed. In total, spectra from 166 samples categorized into 123 subjects and 43 room air measurements could be analyzed for the study. 20 measurements were excluded from the data analysis because of insufficient data. The spectra were statistically evaluated according to the description in the method section, but were not considered individually. The cluster analysis resulted in a total of 139 clusters. According to the results of the linear discriminant analysis, out of the 139 clusters, the PCR positive subjects could be classified from the PCR negative subjects based on the exhaled air samples. The statistic verified a correct classification of 98%. Beyond the classification of PCR positive and PCR negative subjects, an observation of the group classification among the PCR positive subjects took place in comparison to the PCR negative subjects. The group classification was split up into recent infection, which showed symptoms in the time range of 1 to 3 days, and the group of the advanced infection, which showed symptoms in the time range of 4 to 7 days. The statistics of the linear discriminant analysis verified a correct classification of 100% assignment. The conclusion and outlook of this work is that the GC-IMS technique could be a practical application method or diagnostic tool in medical practices for the daily routine by its easy and rapid handling. The used linear discriminant analysis of the data enables a significant statistical verification for the separation between PCR positive and PCR negative tested SARS-CoV-2 subjects. Beyond the study, another confirmational study should be performed to recruit more data for the optimization and standardization to the sampling and the verification of the data methods. By that, for the current outlook, needs to identify by the data more specific of those discriminative VOCs by the determination of matching clusters as potential biomarker to profile SARS-CoV-2 samples. The acknowledgement of the study goes to the IMS teams of the Department Microdiagnostics from the Fraunhofer ITSI in Leipzig, as to the Fraunhofer MEOS cooperating partners in Erfurt. A special acknowledgement goes to the Medical Practice Center of the Drs. Lipp and Am in Leipzig. Thank you very much for your attention.
Brilliant. Thank you so much for the
Hello, thank you.
Hi. Can you hear me okay?
Yes, can you hear me?
I can hear you perfectly. Thank you so
Great.
much for that. So we got a few minutes for questions. I thought I could kick us off. So with the GCIMS system, prior to doing the clinical studies, did you do any characterization work in healthy subjects to try and determine, how many VOCs are detectable on breath compared to background, for instance?
Sadly, we didn't so far. This is one of our future planned steps, also to integrate breath samples from healthy subjects and to compare them, and specifically to see the comparisons between a PCR positive, a really sick subject, to a healthy one. Yeah, but sadly, so far we didn't collect this kind of data.
Okay,
Yeah.
thank you. So you showed in the analysis separation of the clusters, but in the approach, were you able to identify or get tentative identification for some of the specific VOCs that were driving the classifier model?
Not exactly the VOCs, because the created or in the pre-process step of to create the clusters, also to reduce the whole data volume, this cluster were created. And by that, in the statistical part step of using the SPSS, we determined or statistically verified the group classification. Determination of the specific detected VOCs we didn't have so far, but we are also in future work planning to determine by this collected data what kind of VOCs we in particular detected out of these breath samples.
Got it, thank you. So are the features the retention time and the drift time,
Yes.
and then where you have a particular area that you integrate over, so that's classed as a single feature in the analysis. Is that?
Yes, correct. Yeah.
Got it. Thank you. Okay. A kind of broader question related to just the COVID application. So if you look at viral infection, it seems pretty unlikely that you would have unique VOCs being produced. So I was wondering if you could comment on if there was another viral infection, would you expect there to be similar classes of VOCs, and then what that would mean from a classifier performance standpoint?
This is also the one that I showed here in this presentation in the second table of the diagram, the 3D one, because we had this symptom-like COVID symptoms subjects and the real, let's say, COVID patient subjects, and to see if they somehow are showing differences in the VOC profile, what they emitted. And we do think so, even though they show so similar symptoms, they somehow still differentiate from each other. The total or the end answer we still don't have yet, but we hope to achieve this kind of informations, even though they're so similar.
And I know you mentioned that then part of the next step is a prospective validation study. Is that something that's underway at the moment or still being planned?
It's still planned, and we hope that it's going to, that we can do it soon also, to repeat everything and also collect more than these of data.
Got it, thank you.
Yeah.
One of the questions here for the GCIMS device, what parameters did you choose for sampling?
Again, please.
What parameters did you choose for sampling? So I think the question is the sample volume of breath.
We took 10 milliliters of breath by the sample loop.
Got it, thank you.
Yeah.
Next question is, did you consider instead using PCA analysis as a classifying tool?
PCA instead of the LDA, you mean?
Yeah.
Yes, we also discussed that in our group, and we do think also to integrate the PCA analysis also to the LDA, but because of it's not this specified that you can label in particular the group, like ours COVID-like and actual COVID subjects, we chose the LDA, but it still will be a very interesting factor to see the results of the classification by the PCA, which is not specifically labeled. So for future, it's also one of our future working steps to go to the PCA, into the PCA analysis direction also.
Got it, thank you. And the last question is, what analysis softwares did you use? So I'm guessing there was some proprietary software for running instrument and then the separate analysis software.
For the classification, you mean what kind of analysis?
Yeah, so I think the question relates to both. Was there software being used to drive the instrument?
Yes.
Was any data processing happening there, or was that happening in the separate software package?
We used the linear discriminant analysis because of this feature or fact that we were able to label the groups, the cluster, which were pre-processed in the step, and then we analyzed with the linear discriminant analysis. And by that, we were able also to exclude groups which didn't pass the test tolerance, and by that to achieve a more accurate result of the created clusters, and by that to achieve a higher sensitivity towards the classification results.
Brilliant, thank you so much. So thanks again for the presentation and answering.
Thank you very much.
We wish you all the best with the prospective validation study. We'd be very keen to host you again to present those results once that study is done. So thank you.
Thank you very much.